Welcome to episode 94 of Blood and Cancer. I am your host this episode, Nick Andrews. And welcome to episode 94 of Blood and Cancer, the official podcast of MDH Hematology Oncology. I am Nick Andrews, and this week, as you may have guessed, ahead of the Labor Day holiday, we are doing a best of episode with Dr. David Henry and myself. Not recording anything new this week. We thought that because every year ASCO is such a big deal, we would revisit the best of ASCO, and this year particularly important because, as you may have heard, there's a global pandemic, which I suppose is a redundant term. The pandemic forced ASCO and many, many other things to go virtual, but there was also groundbreaking research, a lot of breast cancer stuff that happened, so we've got a great episode for you this week regarding how ASCO 2020 both went as a virtual meeting and some of the great things that were presented there. So the first clip that we have is a conversation between Dr. Henry and myself where we discuss how the meeting went and what are the implications for this going forward. Welcome. That's Dr. David Henry. I'm joined this week, as I always am, by Dr. Henry, and we are literally not even halfway through 2020, and it has already shaped up to be quite the year.
Boy, has it ever, for all of us personally, for all of us professionally, and then, of course, for us as healthcare providers and our patients. So what next? Tornadoes went through Philadelphia today, and yes. So my son, as a young GI doctor in Philadelphia, called me and said, what's next? And, of course, the big upset in the country about what's been happening in protests, as you just mentioned. So it's a lot for people to handle. And to keep us on focus what we're talking about today, we're going to be talking about an interview with Dr. Jack West about lung cancer. But before that, we're recording this just after ASCO, which occurred this past weekend. And I was telling my friends, I'll be in Chicago. They said, "Are you crazy? You can't travel." I said, "I'm not traveling. I'm sitting at home for 4 or 5 hours Saturday and Sunday while your personal life spins around you downstairs." And went to ASCO, which went amazingly well.
Yeah, you give 5 stars.
I can't take my hat off enough to the ASCO staff, Dr. Skip Burris, our outgoing president, too. If you all haven't seen it, you should at least listen to his speech about what it was like. He's a wonderful man, physician, caregiver, and his career in caring for patients, oncology, clinical trials. He goes through all this, was really excellent. And then the meetings, a little bit of unstable. I would tell people I was unstable. They said, "We knew that." I said, no, my connection was unstable. And from time to time, it blipped a little bit because so many people were on, thousands. But then they ironed it out. And now it's all, after the fact, available. So you can all go to Best of the Day. Either of the presentations were just great and are all available online. And to give one shout out that we're going to do this week, Dr. Alan Lyss and I will record after the fact some hot stuff from ASCO that'll probably air in the next week or two.
Yeah, and it give people a chance to kind of digest the meeting and give your guys' thoughts on that. So that'll be great. So here's the question though. So as a journalist and kind of a storyteller and just trying to keep my finger on the pulse of what's going on in oncology, I like the watering holes after the day's sessions. I go to the bars, I go to the restaurants, I kind of do some schmoozing, and I get stuff that we might not necessarily report, but just to kind of check in with the specialty on what's going on. Other than that kind of thing and networking with people maybe you went to med school or did fellowship with, is this, for someone who's just practicing and doesn't maybe want to travel, is this a better way to do ASCO?
I think there's no question that will resonate with many people. I'd already seen, I've been going to ASCO a long time. I went to ASCO when it first started, it was 2 hotels, and now it's major, massive conference centers. So I think over time it has become less relevant for the general practitioner. And I'm a general practitioner. I go because I'm involved with other things, but I can see that it's less useful for the general practitioner who waits for the hot stuff, all this wonderful new stuff presented to be digested, distilled, and how do I put this into place? And he or she could just watch that online and get the best of the day and the discussants to kind of put it all together. I still think there's a reason to go to Chicago, if that's where it's going to be for the next few times, to do all this networking. But I think the online will work for a lot of people going forward.
Yeah, I think that's one of the silver linings we'll take away from the pandemic is that a lot of people have theorized you can do it online, and now that you have to, you think, it's not it's not that big of a deal. But, you and I have been doing this Zoom thing for a while, so we're practiced. So let's talk about our guest. He's a recurring guest, and he knows quite a bit about cancers of the chest. Of course, social media lung cancer influencer Dr. Jack West returns to Blood and Cancer to talk about what's new in lung cancer.
So this was a great interview coming up momentarily. Jack and I talked about some specifics in clinical trials, so maybe 2 months ago in lung cancer. And then with this one, I posed the question to him. I said, well, we have checkpoint inhibitor single agent positive trials. We have checkpoint inhibitor and chemo single or doublet, so 2 or 3 drugs. And then finally, you can even add bevacizumab so that you have 4 drugs and, of course, quite a tab. When would you use which for whom? And he, of course, does a very nice job of decoding that and making it clear where you might want to jump in with everything or just one or some combination thereof. And, of course, do your mutation testing. We touched on that as well.
So Dr. Jack West, of course, you can see back episodes with him by searching in your podcast search bar. They're also linked in the podcast show notes, written by our MD Edge Hematology Oncology editor, Mary Ellen Schneider. And she is a fantastic person who works behind the mic. She's a producer and an executive editor at MD Edge. So she wrote the show notes this week, so you can see that there. Yeah, love to have Jack on. I think Dr. West we should probably check in with him, I say once a quarter, once every about 10 to 15 episodes wouldn't be a bad deal to just check in what's going on in the chest. Yeah. That clip was from episode 73 in your podcast feed. Episode 73. You can find more information about the clips that you hear in this episode in the show notes. We move now to a clip from episode 74. In this episode, Dr. Henry and our guest, Dr. Alan Lyss, discuss 5 abstracts each that they found intriguing or important or perhaps practice-changing. That episode is episode 74 in your feed, so let's get to that clip right now.
Well, the first one that I wanted to bring up, Dave, that I thought was really a significant study was abstract 9500. It was the 3-year update of the Checkmate 227 study. And as you probably know, that study examined a chemo-free regimen of nivolumab plus ipilimumab versus a traditional platinum doublet as first-line treatment for advanced non-small cell lung cancer. And what they did was they treated patients with stage 4 non-small cell, any histology, any PD-L1 expression level, but no known driver mutations. And they treated them with either the novel combination of nivolumab and ipilimumab, a chemo-free regimen, or with traditional platinum doublet chemotherapy. Now, there was a third regimen to which patients were also randomized. It was either nivolumab alone for patients with high PD-L1 expression of at least 1%, or platinum doublet chemotherapy with nivolumab for patients whose PD-L1 expression was less than 1%. There was no crossover in this trial, and the primary endpoint was overall survival. But as you might guess, over 75% of the patients who did not receive a checkpoint inhibitor as part of their randomized therapy eventually received one. And what they found at first blush, I thought was an incredible win for nivolumab plus ipilimumab. For the patients with PD-L1 expression of at least 1%, the 3-year overall survival rates were 33% for nivolumab plus ipi, in comparison with 22% in the chemotherapy arm. When did you and I ever think we'd be talking about 3-year survival with 3% for metastatic non-small cell lung cancer? The nivo arm, as you might guess, was intermediate between those two. But the interesting thing was that if a patient had a CR or PR in the first 6 months of their therapy, almost 40% of the nivo plus ipi patients remained in response at 3 years, in comparison with, as you might expect, only 4% of patients in the chemotherapy arm. So 10 times more in the nivo plus ipi arm. Now, if a patient's PD-L1 expression was 0, the 3-year overall survival with nivo plus ipi also wasn't too bad. It was 34%, but it was only 15% for chemo alone. And again, it was intermediate for nivo plus chemo. And once again, if a patient had a response by 6 months, even for patients with PD-L1 expression of 0, about a third of them on nivo plus ipi were still in response at 3 years, in comparison with 0% for the chemo alone group. And what I felt this meant to us and to our patients was not as much as you might think at first blush for two reasons. One was toxicity and the other was time to respond. Anybody who's given Nivo plus Ipi knows this is not for the faint-hearted. Grade 4 toxicities were seen in about a third of patients. About twice as many of them stopped their treatment prematurely as stopped chemotherapy for toxicity. But the other thing that the discussant brought up that I thought was really interesting is that if a patient is that for Nivo plus Ipi, if there was clinical deterioration and death within the first 6 months, there were more of those on Nivo plus Ipi than there was with chemotherapy alone. It took about 6 months for the curves to cross. And I personally felt the bottom line here is that there's still room for clinical judgment. Disease-associated symptoms, burden of disease, performance status, comorbid conditions, and perhaps even advanced tumor genomics, like having the presence of a molecular profile that might code for resistance to an immune checkpoint inhibitor. I think all those things still have to enter into the equation of whether a chemo-free regimen is appropriate for a given patient.
Yeah, and we actually interviewed Jack West a week or so ago, and this is prior to ASCO, talked about exactly that. Tailor-make to your patient, now that we have so much choice, checkpoint inhibitor plus chemo, checkpoint inhibitor only, et cetera. Well, I saw that as well, thought that was really interesting and a little more in the side effect profile, but doable and considerable. Well, let me go then to my first one, Abstract 501. So I'll read the title. 3-year follow-up of neoadjuvant chemotherapy with or without anthracyclines in the presence of dual HER2 blockade for HER2-positive breast cancer, the TRAIN-2 study, a randomized phase 3 trial. The presenter was Anna van der Voort, and she's from the Netherlands. And so the authors studied here neoadjuvant therapy, trastuzumab, pertuzumab, received by all, HER2-positive disease, locally advanced, stage 2-3, and they then randomized to get one of 2 different chemotherapy arms, either 3 cycles of FEC, so the epirubicin anthracycline, and 6 cycles paclitaxel carbo, so 3 and 6 in the first arm with 3 of anthracycline, or 9 cycles of paclitaxel carbo only, and then that followed with a year of trastuzumab, as we typically do for HER2 positive. Radiation may have been in there if you had lumpectomy, endocrine therapy in there if you were hormone positive. So what did they find? They had a lot of patients in the international trial, 438 patients, randomized, and they found a 3-year event-free survival of 92 in the experimental arm and 93, or so I should say in the anthracycline arm and 93 in the non-anthracycline arm, event-free survival. So no difference. And the overall survival estimates, so OS, comparable 97 versus 98. So what's their point? They said, given this, and I underline this in a HER2 population, although it's being sorted out in non-HER2 populations, a physician could choose a non-anthracycline-containing regimen and avoid the acute and chronic toxicities. I think there's more nausea and vomiting. We always worry about the heart. We always worry about downstream bone marrow effects. So do we really need anthracyclines in this neoadjuvant, let alone adjuvant setting? So this was in the HER2 population. So I thought that was one more piece in the non-anthracycline story of whether or not you need them. So interesting. Back to you.
I felt the same way. Mm-hmm. And as you and I learned many years ago, you can't be afraid to unlearn something that you thought you already knew. You know, if drugs A plus B plus C are better than A plus B, and if C is such a terrific drug, it might be possible to ask whether you needed A at all.
Yeah.
And it seems like that's what they did in this study.
Agree. Yeah, interesting. So that story continues.
The next one I wanted to bring up was actually one of the plenary session talks. It was LBA 5. This was the ADORA trial, and it kind of keeps us in the theme of non-small cell lung cancer for my presentation. And this was a study of osimertinib as adjuvant treatment for patients with EGFR mutated non-small cell who had had a complete tumor resection. And what they did in this trial is they randomly assigned patients with stage 1B to 3A EGFR mutated non-small cell lung cancer who had come through potentially curative surgery to either adjuvant osimertinib at standard doses or an identical placebo for 3 years. Now, if patients required adjuvant chemotherapy, they were allowed to receive it for whatever stage they had after a discussion with their oncologist. But the osimertinib or placebo began after the conclusion of the adjuvant chemotherapy in that case. And they randomized almost 700 patients on this trial. They had co-primary endpoints of disease-free and overall survival. But the Data Safety Monitoring Board recommended stopping the trial 2 years earlier than planned because of dramatic differences in disease-free survival. And what they found was that for stage 2 to 3A patients, the disease-free survival hazard rate was, get this, 0.17 in favor of osimertinib.
Wow.
The curve separated early and progressively so that by 3 years, the disease-free survival for osimertinib was 80% in comparison with 28% for placebo.
And tell me again, these are mutated driver mutations or not?
EGFR mutated driver mutations.
Got it, okay.
Right. For stage 1B patients, the disease-free hazard ratio was 0.50, so not nearly as impressive, but, 0.50 is a hazard rate that in any other context would be viewed as a huge win. So at 2 years for the stage 1B patients, 87% of those on osimertinib were alive, free of disease. Alive, free of disease, compared to 70% for the placebo patients. Now, for any adjuvant treatment, obviously toxicity is a huge issue, but it wasn't much of a serious issue with osimertinib. The patients stayed on osimertinib actually longer than they stayed on placebo, and there were no adverse events leading to death. And personally, what I think this means for our patients and for us is that once this drug gets approved for this indication, we ought to be using it, despite we don't yet know what its overall survival impact might be and whether it's curative. You and I had a mentor 40 years ago, Dr. John Glick, who taught me very poignantly that disease-free survival is an endpoint in its own right. Patients generally have better quality of life before relapse than afterwards. So DFS should not be de-emphasized here.
Couldn't agree more. Sounds very practice-changing, for sure. Wow.
All stages of non-small cell lung cancer, we've got to be testing for a sensitizing EGFR mutation because there's now a therapeutic option that could benefit.
And that was really hit throughout the meeting. We've got to find, because we'll probably hit 100% one of these days, but now it's about a third. When I remember a few years ago, it was like 5%, so we need to look.
Absolutely.
So let me then go to next abstract on my hit parade of 5, number 2, abstract number 1000, easy to remember. Keynote 355 study, titled A Randomized Double-Blind Phase 3 Study of Chemotherapy with or Without Pembrolizumab for Previously Untreated Locally Recurrent Inoperable or Metastatic Triple-Negative Breast Cancer. So all comers of triple-negative breast cancer. Dr. Cortez presented, and it's an international trial. So I've been fascinated by checkpoint inhibitors showing lots of promise, and I'll mention a new drug approval in a moment, in treating triple-negative breast cancer. And so this study extends that theme. It was a 2-to-1 randomization, and the chemotherapy was either nab-paclitaxel or paclitaxel or gemcitabine carboplatin, so 3 kinds of chemotherapy. And they randomized 2-to-1 to be with or without pembrolizumab. And the response rates, especially in those as common theme, enriched in higher PD-L1 expression, something they call the CPS score, combined positive score, greater than or equal to 10 was especially impressive. And I just give some quick hazard ratios. If you're CPS 10 or more, you're a median PFS, progression-free survival, 9.7 versus 5.6 months. These are triple-negative breast cancers. CPS 1 or more, 7.6 versus 5.6. So a very positive trial, very statistically significant. And here's something now in the triple-negative breast cancer space. And I wanted to mention a recent drug approval, a trade name Trodelvy, which is sacituzumab govitecan. I remember seeing this at ASCO a couple years ago when they presented. This is a drug-antibody conjugate. The triple-negative breast cancers are highly enriched in some marker called TROP2, TROP2. And so the antibody part of this drug-antibody conjugate goes for the TROP2, and it comes loaded with a topo-1 isomerase inhibitor, SN38, also known as govitecan. So a neat delivery system. I've just started using it in a couple of my triple-negative metastatic breast cancer patients. So here's abstract 1000 showing anything from early to metastatic, and the combination with a checkpoint inhibitor works better than without in the chemo regimen. And then we, of course, have this drug antibody conjugate. Really interesting developments in a very difficult cancer.
And finally today, we're going to wrap up with our preview of the virtual meeting, which is something that I thought would be an interesting way to wrap up episode 94 of Blood and Cancer, because we did this preview with Dr. Skip Burris on how the meeting was going to go and what the plan was for the plenary session and what the plan was for different educational sections. And I think that the best way to end this best of episode is to look back on what the preview was going to be and what the implications were. So this is Dr. David Henry and Dr. Skip Burris.
You wonder, I'm sure there'll be a post-mortem, so to speak, after this is all done this summer of what worked and you might keep going forward. It's certainly taking a lot of time and effort and technology, but maybe that's something for those who can attend, could watch posters and presentations in a future format.
I think you're right. Everything about the way we're practicing medicine, attending meetings, interacting is likely to be different. It's going to be like every other event where we talk about pre or post the Gulf War or 9/11.
Yeah.
I think we'll be about pre and post the pandemic. My patients, nervous at first, have loved the telehealth visits. I was dreading having my face on webcast all the time, and now it turns out that it's just part of life now. I think similarly, Cliff Hudis, ASCO CEO, and myself have discussed this. How the ASCO meeting looks going forward will probably change. People are going to like this ability to go back and review the data and how we present it. So I think you're right. We're going to see a different ASCO meeting going forward, and we'll take the best that we learn from this meeting and apply it going forward.
Indeed, making lemonade out of lemons. Wow, what an amazing turn of events that'll give us something new and different. Well, lastly, I thought we might, I know ASCO is doing something called Covet Cancer Registry. Can you explain what that is and how that'll work and how that might help us?
Yes, thank you for bringing that up, David. So we have first received at ASCO just a number of questions that came in from both patients through the cancer.net side and from our members through the ASCO.org about COVID-19, concerns about should patients be treated, how do we continue on, the phrase stay safe but stay the course. Cancer is so different. We can't shut down like we have done with regard to some of the joint replacement surgeries and valve replacement surgeries in the cardiovascular arena. So ASCO decided the best we could do, we put out the frequently asked questions and got some experts to comment, but really to begin to aggregate real-time data and learn from this. So under the leadership of Dr. Rich Shulsky, who heads Centra, ASCO's Center for Research and Analytics, we launched the ASCO Registry. We reached out to our pharma biotech colleagues and through Conquer Cancer and received some support so a special thanks there for those companies who've helped put this up with our manpower. And this is an idea to not just get a snapshot of data, but to actually get longitudinal data. And with the funding that's coming in, we're hoping that we can continue to have the sites receive some funding to continue both collecting the baseline and follow-up data about how the virus is impacting cancer care and patient outcomes. I think we're going to learn a lot from just basic demographics, but we're going to see and be able to track various groups of patients and how they perform. There's been stories out of China and other earlier hotspots, Italy and the like, about lung cancer patients versus not, older patients versus those who are younger, those who are on immunotherapy. We plan on having real-time data releases, so we're going to deliver periodic reports to the cancer community and the broader public on key learnings, try to have it as a real-time educational tool just as soon as we learn something about the COVID-19 epidemic and how it's affecting cancer care to get that information out. So some of the early things we're already taking note of is the rapid implementation of telemedicine, and seeing some of the treatment modifications and delays. So exciting. There's a number of registries that are out there, and we're partnering with other societies to make sure the data is aggregated. But this, I think, will be unique in the fact that it's going to have a longitudinal aspect to it with some longer-term follow-up. And it will just be a great learning tool for our oncologists and hopefully an educational tool for other providers and our patients.
Well, really incredible. And if people want to know more about this or how to participate or generate or give data, it's on the webpage. How would they find out more about it?
Right. So great question. On ASCO.org, there is a link that takes you straight into the registry. We've got a number of practices that have already signed up. So there's great information there about it. It's actually fairly straightforward process in terms of how the various practices can participate. And actually, there's a wealth of information that's on the ASCO website, ASCO.org, about COVID-19 and some of the things that are going on and some of the educational tools. And then for your patients, Dr. Mary Markham is one of our cancer communication leaders, and she has been having a blog about coronavirus, COVID-19, and patients with cancer. That's on cancer.net. So I encourage any of our listeners who have patients with questions. That's a very friendly website, cancer.net, and a great place to get some information.
Well, Skip, I think we've covered everything I wanted to ask, unless I missed asking you something about what you'd like our members to know. I really thank you for taking the time to talk to us. And on our Blood and Cancer podcast, to remind our listeners, we've been speaking to our current president, who will certainly go down in memory as one of the most unusual years, and thanks to his leadership, one of the best outcomes that could be happening for ASCO. Dr. Skip Burris, who is Chief Medical Officer at Sarah Cannon and our current reigning ASCO president. Skip, thanks so much, and good luck to you.
And that's a wrap on episode 94. This best of edition of Blood and Cancer was produced by yours truly, Nick Andrews, along with MD Edge editor Jennifer Smith, Medscape editor Lauren Gravitz. All MD Edge podcasts are produced by executive editor Kathy Skarbeck and Medscape senior vice president of editorial Dr. Ivan Aransky. I'm your host, your audio engineer, and of course your audio editor. You're listening to Blood and Cancer